Hello everyone, welcome back to our channel. So today we are going to talk about the shuttle vector. So what is a shuttle vector? So let's dive into this shuttle vector. So this shuttle vector is basically a plasmid engineered vector so that it can propagate in two different host species like the bacteria as well as the yeast species. Now we have here the yeast shuttle vector. As the main components here are the it contains the bacterial origin of replication as well as the yeast origin of replication so that it can replicate in bacteria as well as in the yeast, and we need the yeast selectable marker as well as the bacterial selectable marker. So this is the shuttle vector. So we can say that these shuttle vectors, they contain the both types of the components, from the bacterial origin components and the yeast components. So from the bacteria, we have the bacterial origin of replication, we have the selectable marker for the bacterial selection. We have here also, in case of the yeast, origin of replication as well as the yeast selectable marker. And then we have here the centromeric region for the yeast. And we have also the autonomous replicating sequence so that it can replicate in the yeast. So this is the shuttle vector. Now what are its advantages and disadvantages of the shuttle vector? So here the advantage is that the main advantage of these vectors is that they can be manipulated and they can be grown in E. coli, and then they can be used in a system which is more difficult or slower to use, for example the yeast. Whereas the disadvantage is that the carrying capacity is low. So here we have the low carrying capacity, but the advantage is that this can be manipulated and this can be grown in E. coli, and this can be then used in case of the yeast also. So this is the advantage of this shuttle vector.